Today, we're diving deep into the science behind a truly remarkable field, non-invasive brain stimulation. We're going to explore exactly how these tools work, right down at the cellular and molecular level, to treat incredibly complex conditions like depression.
Okay, so here's our roadmap. First, we'll establish why these technologies are so desperately needed. Then, we'll get a look at the tools themselves. We'll explore the core molecular mechanism, which revolves around the protein called BDNF. After that, we'll review the hard evidence for each technique, tackle the challenges of bringing this science from the lab into the clinic, and then wrap it all up with the key take-home message.
So first, let's set the stage. Major depressive disorder, or MDD, is a leading cause of disability across the globe. But here's the tough reality. For a huge number of patients, our standard treatments just don't work well enough.
Just think about that number for a second. Nearly a third. That represents a massive treatment gap for an absolutely debilitating condition. And it's this urgent unmet need that's really fueling the worldwide search for better therapies, like the non-invasive brain stimulation techniques we're about to explore.
So let's get into the techniques themselves. The term NIBS, that's non-invasive brain stimulation, is really an umbrella term for a whole range of incredible technologies. What they all have in common is that they're designed to modulate, or kind of fine-tune brain activity safely from right outside the skull.
This table really maps out the major players in the field. You've got techniques that use magnetic fields, like TMS, others that use gentle electrical currents, like tDCS, and even well-established methods like ECT. Now, while each one has its own primary mechanism, from boosting synaptic plasticity to modulating neurotransmitters, the really fascinating part is how they all seem to converge on a single, central molecular story.
And that brings us right to the heart of the matter, to the core molecular story. You see, despite all their differences, these technologies seem to channel their therapeutic effects through a critical family of proteins called neurotrophins. These are the key players in the brain's ability to change and adapt, what we call neuroplasticity.
And right here on this slide is our central player, brain-derived neurotrophic factor, or BDNF for short. Honestly, the best way to think of it is like a powerful fertilizer for your brain. It promotes the survival, the growth, and the connectivity of your neurons, all the things that are so often compromised in depression.
But here's where it gets nuanced. It's not just about having more BDNF. It's about the balance between two different forms. On the left, you have mature BDNF. That's the good stuff. It's protective and promotes growth. But on the right, you have its precursor, pro-BDNF, which can actually be harmful, triggering inflammation and even cell death. In MDD, this balance is often tipped towards the harmful side. So the goal of these therapies isn't just to crank up total BDNF, but to flip this molecular switch back, boosting the beneficial, mature form and restoring a healthy balance.
And this slide shows the incredible chain reaction that happens next. Once that mature BDNF binds to its receptor, it kicks off a whole cascade of signals inside the neuron. And we're not just talking about a fleeting chemical change here. This process activates pathways that literally regulate protein synthesis, leading to real physical changes, like the growth of new dendritic spines. This is neuroplasticity in action. This is the structural foundation for long-term therapeutic effects.
Okay, so now that we get the whole BDNF hypothesis, let's really dig into the evidence. What do the clinical trials and lab studies actually show? How do these different NIBS techniques really impact this critical system?
Well, the data for repetitive TMS, or rTMS, is pretty compelling. The studies you see here consistently show that high-frequency protocols lead to a significant increase in BDNF levels in the blood. And here's the critical part. This molecular change directly correlates with positive antidepressant outcomes in patients. That's a powerful direct link between the intervention, the biomarker, and the clinical result.
Now with tDCS, the picture gets a little more complex. As you can see, the clinical outcomes are positive. The patients are getting better. But these studies didn't consistently find a change in those neurotrophin levels we were talking about. This is a fascinating puzzle. It suggests that while tDCS is clearly effective, it might be working through different, or maybe just more subtle, mechanisms that our current blood markers aren't sensitive enough to detect.
Okay, next up is ECT. Now, this is one of the most effective treatments we have for severe, treatment-resistant depression, and the evidence here is incredibly robust and consistent. Recent studies directly link its powerful clinical effects to significant increases in BDNF,
And that study by Z and colleagues is especially groundbreaking. It showed direct evidence of increased neurogenesis, the actual birth of new neurons in humans following treatment. Just amazing.
So this all leads to a really critical question, right? We have all this compelling molecular evidence, especially for BDNF. So why aren't these NIBS treatments a perfect, predictable cure for every single person? Why do the outcomes still vary so much?
Let's unpack the translational hurdles, the things that create this gap between our scientific understanding and the reality of everyday clinical practice.
You know, a few key factors really complicate the picture. First, our animal models, while incredibly useful, just don't capture the full complexity of the human skull and brain. Second, the BDNF we measure in blood is an indirect biomarker. It's like an echo of what's happening in specific brain circuits, not a direct measurement. Third, our own genetics play a huge role. There's a common variation in the BDNF gene itself, known as Val66MAT, that can significantly influence how a person responds to treatment. And finally, there's a lack of standardized protocols across clinics, which makes it really hard to compare results and optimize care for everyone.
So, after this tour through an incredibly complex and exciting landscape, what is the single most important message you should take away from all this research?
The key takeaway is really this. Non-invasive brain stimulation is so effective because it taps into the brain's fundamental capacity for change, its neuroplasticity, and the BDNF system seems to be a master regulator of that process. These aren't simple on-off switches. They're sophisticated tools that influence a complex biological web. The future of mental health treatment isn't just about finding the next pill. It's about leveraging these deep molecular insights to develop truly personalized therapies, using biomarkers to guide precise, individualized neuromodulation.